Hi guys, it's Miss Loudon again. Thank you so much for watching my next video, which is about coordination and regulation. So detecting all of these different changes in our environment and coordinating a response so every single cell in our body is able to counteract and balance that change so we can maintain homeostasis. This topic might look familiar to some of you because we did cover some of it in Year 11. I'm going to bring up the stimulus response model that we covered in Year 11, and I'm also going to mention temperature regulation. So hopefully some of this stuff feels quite familiar to you. Of course, I appreciate this stuff at the bottom here might not, but we'll deal with that in time. I also can't wait to share some of the stories and medical trials around learning about some of these different signaling molecules. Okay, so before we get started, we need to remember that we have an internal environment and an external environment. So my internal environment is everything underneath my skin. It's the fluid between my cells. And the external environment is obviously what's outside me. So, for example, at the moment, it's raining. So that's my external environment. And what's really important is the concentration of different substances inside my body need to be maintained at very, very specific levels, otherwise, that will lead to dysfunction. So, the way that organisms are able to do that is by constantly trying to move substances across the membrane to maintain a balance of substances within the cell and within the body. So, what I mean by this is that we have intracellular fluid, so inside the cells of body tissues and inside red blood cells. So it's the intracellular fluid. And we also have extracellular fluid. It's still in my body, right? But the blood plasma here, for example, and the tissue fluid is outside the cell, inside my body, but outside the cell. That's why we say it's extracellular. And this question here asks, how are these fluids separated? And this is really important. They're separated by plasma membranes. So what we see on the outside of this cell here, and also outside the body cells are plasma membranes, fluid mosaic model plasma membranes. So they have receptors, they have protein channels, and they're able to select which molecules are able to enter the cell and control which molecules are leaving the cell as well. So what we're aiming for when we're talking about coordination and regulation is we're talking about maintaining homeostasis. And this is really the first thing that I think we need to make sure we have perfectly written in our notes, and that is our textbook definition of homeostasis, which is the maintenance of a relatively stable internal environment of cells within a small range of tolerance in the face of changes in the external environment. So even though the external environment might be changing, we need to maintain homeostasis. And if you think about the different molecules that we need to keep in balance, glucose, oxygen, carbon dioxide in particular, even just a tiny bit more or less of these molecules outside our normal tolerance range, in carbon dioxide, for example, you're going to lose consciousness. So we really need to keep these different molecules in check. So the next part of what we're going to learn is how we actually do keep these balanced. So we're going to talk about different homeostatic mechanisms. All of this is getting to where we understand how we're going to, for example, take glucose from the external environment into the internal environment or back the other way, and also make sure we're removing our waste products. So we're constantly exchanging molecules from inside the cell to outside the cell to make sure that we are able to maintain homeostasis. The way that this is achieved is through the stimulus response model, and this is referred to in the study design, and the language that I use here, so stimulus, receptor, effector, response, and control center, are really important. You'll see the definitions on slide 9, but I'll just go back to what I was talking about. So the stimulus is any change in the environment, and that can be an internal change or it can be an external change. So let's just use an example. The stimulus, in the example that happened to me recently when I was staying with my family in Canberra, is that I heard a possum scratching in the wall of my childhood bedroom in my house. So I was hearing scratching. The receptor was just the receptors in my ears that heard the sound. And the control center its definition is whatever mechanism there is for the receptor to get a response. In this case, it's my brain. It interprets the sound. I decide that it's clearly a murderer. The control center, my brain, is going to try and get my body to respond to that threat. And the way it's going to do that is it's going to transduce a response. So it's going to transduce a signal, maybe through nerves and hormones. I'll focus on the hormone here. So it's going to trigger a surge of adrenaline that's going to target lots of different effectors in my body, but one effector, which is just a thing that produces a response, is probably going to be my heart, and the response is I am going to increase my heart rate. My heart is going to be thumping in my chest, ready to pump blood all around my body to prepare for my flight or fight response with this murderer. So that's basically how the stimulus response model works. The stimulus is the scratching in the walls, and the response is the increased blood flow around my body, increased heart rate. So it might be worth pausing and taking some time to draw a diagram of this. So you could have this language and add in more of this information that you see here, and also add in these really important definitions. And seeing that we've got a stimulus, which is going to be detected by receptors. We've got different communication systems to transduce the signal to our effectors, and that will elicit a response. Most of what we're going to be doing in the next few lessons is around how we trigger from the communication systems, how we trigger a response inside a cell. But we need to know the whole loop here. Now, the important part, which is different to this picture, is it's actually in a circle. And the reason is because the stimulus response model works best when you can check the stimulus, see if it's still there, and adjust your response. So obviously, if I've heard scratching in the walls, my response has been to increase the blood flow around my body. My heart's beating really fast. I don't want that to go on forever. So I'm hoping there's some kind of mechanism in my body that's going to realize, oh no, it was just a possum. And I'm going to have my adrenaline switching off and therefore my heart rate lowering. Okay, so I've mentioned that you need to have these definitions written down somewhere. Okay, so we can't just have the response going on forever. We have to be able to control the response in the stimulus response model. Because once the signal's been responded to, we really need to switch it off. It's not healthy, and we're not going to be able to maintain homeostasis if we absolutely overcorrect every time we detect a different signal. So there are 3 different strategies the cell can use to switch off the signal. You can disrupt the signal transduction pathway, and we'll come to talk about that. You can remove the original stimulus, so that's what happens when you realise that it's a possum, not a murderer. But the one that I want to talk to now is actually responding in a way that will alter the original signal or reduce the original signal, so you're not going to keep responding. Okay, and this is in the form of a negative feedback loop. It is a feedback loop used in 99.9% of cases where we talk about detecting and responding. And we've got a really important definition of a negative feedback loop here. So a negative feedback loop is just where the response causes the stimulus to go back in the opposite direction. So here I'm going to use the example of temperature regulation that we covered in Year 11. So the example I'll use is, maybe it's cold weather. You walk into a room, the heating's on, it was initially feeling quite pleasant, but now it's starting to feel quite hot. And you might remember that we have temperature sensors, so misalignment detectors in our hypothalamus, and it actually measures our blood temperature. And the sensors really want our blood temperature to be 37 degrees, and unfortunately, so this is the signal that my hypothalamus is detecting is that actually I'm quite warm. In fact, I'm probably around 37.5, 37.6 degrees. I'm starting to get too hot. So my temperature is above optimal. That's going to trigger a whole range of responses. So one of the effectors, for example, could be my sweat glands to produce sweat, and hopefully that sweat production will cause it to evaporate. That's going to lower my body temperature, and that should lower my temperature. Now, if it goes too far, because the aim is to lower the temperature, decrease it, if it actually goes too far and it's below optimal, then that's going to again trigger the effector to change the response. So for example, it could just be to switch the sweating off, or if it's really gone too far, it could be to actually counteract that, maybe through shivering or something to increase my temperature. So the whole idea of a negative feedback loop, which we should write down, is to make sure that the response makes the stimulus go back in the opposite direction. So we're counteracting the change that we've detected. If I've detected low temperature, then my effector will respond to raise temperature. If I've detected that my temperature is too high, my effector will respond to lower that temperature until it's perfect. But it's never perfect, so it's always working. Another really useful example, it comes up in exams all the time, is the regulation of blood glucose or blood sugar. So this is also using a negative feedback loop. So you might have heard that blood sugar is controlled by two different hormones. So you've got insulin, which is released by beta cells, and it causes our blood to give up its glucose. So it's going to take up the glucose from the blood, reducing the blood sugar levels, and it stores that sugar or the glucose in tissues in the form of glycogen. And the other hormone is glucagon, and that does the opposite. It's released by the alpha cells in the pancreas, and it's going to cause glycogen in the liver to be broken down into glucose, and that glucose will then be released into the blood. So glucagon is going to raise blood sugar, and insulin is going to reduce blood sugar levels. Okay, so let's take a look at this stimulus response model negative feedback loop that we have in front of us. So the sensor, or the thing that is detecting the stimulus here, is going to be these Langerhans cells. So it's going to be the cells in the pancreas. And that, for example, maybe these cells are going to detect it's been a while since I've eaten and my blood glucose levels are actually below normal. This is going to trigger a release of this hormone glucagon, which is going to hopefully get my effector, in this case the liver, to respond. And the response it's going to trigger is it's going to convert the glycogen in the liver to glucose that should go into my bloodstream, and with any luck, my blood glucose level will rise. Similarly, another hormone that could act in this way is insulin. If you reduce the amount of insulin, then you've got less glucose being absorbed by the cells. These, like muscle cells, for example, are the effector here. And hopefully my blood glucose level rises. And if we follow that through, if it rises just a bit over what it should be, so it's now above the normal range, I've counteracted that low blood glucose and now it's just a little bit too high. So again, that's going to be detected by the Langerhans cells in the pancreas. That's going to cause the alpha cells in my pancreas to actually lower the amount of glucagon going around, which is then going to act on the effector, which in this case is going to move the glucose from the bloodstream back into the liver. And also the same thing will happen with the release of insulin, which is going to cause my effector, so for example these muscle cells, to absorb more glucose. Hopefully my blood glucose level falls, and it's a never-ending cycle. This obviously happens in a non-diabetic because if it was a diabetic and they didn't have any insulin being produced by their beta pancreatic cells, obviously this feedback loop wouldn't work. And that's why we hear of people going hyperglycemic when they have type 1 diabetes. Okay. The next example of a feedback loop in the stimulus response model is much rarer, especially in humans, and it's a positive feedback loop. So here, instead of the response hopefully making the original signal go in the opposite direction, in a positive feedback loop, you want the response to go in the same direction as the initial change it's detected. So I'll explain what that means. And the best explanation is actually the example of the release of this feel-good chemical oxytocin, which is released during childbirth. Now, oxytocin does two things. It really makes the mother experience much less pain, feel more positive about the whole experience. But it also assists with contractions and the cervix stretching so the baby can actually be born. Okay, so what happens when you get these initial contractions and the baby's head starts to push downwards? That actually triggers a release of oxytocin, which in turn triggers more contractions and more stretching, which in turn releases more oxytocin. So we're actually magnifying the signal each time that it's going through. And if you think about it, this is a great thing to happen because it's going to really help childbirth occur. We wouldn't want to balance out and make sure childbirth can go for as long as possible. We have other examples in the plant world. For example, the ripening of tomatoes or the ripening of bananas works in a really similar way. This brings us to the end of the lesson. And what I want you to be able to do by the end of this is be able to put these different principles of communication in order. So we've got detection, a response, switching off, producing a signal, and transduction of the signal to the target cells. And I hope that you're able to put these in order. Can you do it? Can you do it? Well, this is the answer. And I really look forward to talking about different signaling molecules with you at our next lesson, which I can't believe is Tuesday. Thank you so much for watching this, and I'll see you soon. Bye.